This video is going to cover section 13.2, which looks at the structure of the kidney in a little bit more detail. And the kidney's blood supply is supported by many branches of the renal vein and renal artery. And the kidney has three regions. It has the renal cortex, which is the outer granulated layer. It has the renal medulla, which contains cone-shaped tissue masses called renal pyramids. We have the renal pelvis as well, which is the central space that's continuous with the ureter. Here you can see some various diagrams of cross-section of the kidney. And the one on the right would actually be a photo of an actual specimen. Again, a cross-section of it. So you can see the three regions of the kidney in this, with the outer renal cortex, and then the renal pyramids in the renal medulla, and then the inner renal pelvis. The kidney is composed of over 1 million nephrons, also known as the renal or kidney tubules. And you can see that they extend into all three regions of the kidney itself, right from the cortex into the renal medulla and the renal pelvis. So it's basically the structure of the nephrons themselves which determine the structure of the kidney into its three regions. The blood supply in a nephron, each nephron has its own blood supply from the renal artery. An afferent arteriole leads to the glomerulus, which is a knot of capillaries inside the glomerular capsule, which you can see labeled on the diagram there. And blood leaving the glomerulus then enters the efferent arteriole, which takes blood to the peritubular capillary network before exiting through the renal vein. And you can see those structures as well on that diagram. So the glomerular capsule, it was formerly known as the Bowman's capsule. We're trying to get away from proper names for things and refer to them in the names that have a little bit more meaning. So the glomerular capsule, it has an inner layer which is composed of cells called podocytes. And the spaces between the podocytes, they allow small molecules from the glomerulus to enter the glomerular capsule. And again, you can see that labeled on the diagram there. The proximal convoluted tubule then is lined with cuboidal epithelial cells that have packed microvilli to increase the surface area for reabsorption. And they also contain several mitochondria to carry out some active reabsorption of substances as well. We have the loop of Henle, or Henle's loop, and this is also known as the loop of the nephron, which will probably get renamed in that way, the loop of the nephron. It consists of a descending limb and an ascending limb. And these are lined with simple squamous epithelial cells. The distal convoluted tubule, it's composed of a cuboidal epithelial cells that lack microvilli, but they have many mitochondria then. So that tells you then that the purpose for this area then is for active reabsorption. So it helps move molecules from the blood then into the tubule. So the distal convoluted tubules of many nephrons enter one collecting duct, and the collecting ducts then carry urine to the renal pelvis. And here you can see all those structures, as well as we have some microscope sections there, as well as the top. The top right may be an electron micrograph. I'm not sure. It may just simply be an illustration. It's kind of hard to tell. But you can see how the structures look. Obviously, when you take a cross section, depending on how the different structures are oriented, that's going to determine how they how they're observed in cross section. So it's sometimes difficult to know exactly what is what when you take an electron, or when you take a slice of tissue and look at it under the microscope. So with urine formation then, urine formation is divided into the following processes. So we first have a pressure filtration, which occurs in the glomerular capsule. And so this is where we filter water, salts, nutrients, and wastes. Then we have selective reabsorption, so nutrient and salt molecules are actively reabsorbed from the convoluted tubules into the blood of the peritubular capillary network. And then we have tubular excretion, which certain molecules are actively secreted from the blood into the peritubular capillary network into the convoluted tubules. So we'll first look at pressure filtration then. And this occurs in the glomerulus, as mentioned. This is in this diagram. You see all three of those processes and where they kind of take place. So we'll look at each one of those in a little bit more detail, starting with the pressure filtration. So it occurs when blood enters the afferent arteriole and the glomerulus. So just simply due to blood pressure, water and small molecules, they move from the glomerulus to the inside of the glomerular capsule. And these filterable blood components form the glomerular filtrate. And you can see the things that are filtered there, as well as things that are not filterable in this case, like for example formed elements, blood cells and platelets, as well as plasma proteins. So the things that are filterable would be water, nitrogenous waste, nutrients, and salts. With selective reabsorption, nutrients and ions from the glomerular filtrate in the convoluted tubules, they're reabsorbed into the blood of the peritubular capillary network. So some examples of this would be sodium ions are actively reabsorbed into the blood, and then chloride ions, they follow along passively in order to maintain the electrical charge. So this reabsorption of salt, it results in water moving passively from the tubule into the blood. Glucose and amino acids are actively reabsorbed into the blood as well, almost exclusively at the proximal convoluted tubule. So here you can see how much of each of these substances are filtered per day and how much is reabsorbed. So in an average day, we filter about 180 liters of water, and we excrete about 1.8 liters, so that means that 99% of that is reabsorbed. With sodium, we filter about 630 grams, and we only excrete about 3.2 grams of that. So the sodium gets reabsorbed, about 99.5% of it. Glucose, we don't want to lose any glucose, so we reabsorb 100% of that. Urea, we don't actually excrete all of the urea, so about 44% of that is actually reabsorbed. So with selective reabsorption, the glomerular filtrate that enters the proximal convoluted tubule, it's divided into two portions. So we have reabsorbed filtrate components are reabsorbed from the tubule into blood, and then we have non-reabsorbed filtrate components. These continue to pass through the nephron to be processed into urine. So it becomes the tubular fluid that enters the loop of Henle. And you can see in the chart here. So reabsorbed filtrate components would be most of the water, nutrients, and required salts. Some of the non-reabsorbed filtrate components would be some water is excreted, most nitrogenous waste is, and excess salts. Tubular excretion is another way that substances are moved from blood in the peritubular capillary network and added to the tubular fluid. So hydrogen ions, potassium ions, creatinine, and certain drugs are removed by active transport from the blood in the peritubular capillary network into the convoluted tubules. So the resulting urine then ultimately is going to contain substances that have undergone glomerular filtration but have not been reabsorbed, and substances that have undergone tubular excretion. So that's a little bit more on the detail of the structure and the function of the kidneys, and we will continue looking at the excretory system.